Good morning, Vietnam. Okay. Mine. You like Gollum? You're precious. Okay, that's everything. Very good. It's time to start. Well, welcome back, everyone. Hope you guys went to bed early enough to get that extra hour sleep. Or not extra hour sleep, you didn't lose an hour because you didn't take into account the time change. That spring time change for me is never fun. And I'm not a big fan of waking up in the dark. So not a fan of waking up early in the first place. Waking up early when it's really dark, not a fan. Not a fan. So, but I suppose the longer days of longer daylight period is fun. So there we are. Okay, so we are going to finish our discussion of genetic counseling. Then we'll get into neurodegenerative diseases. We'll look predominantly at Alzheimer's disease, but we'll also look at Huntington's chorea. And nursing care when it comes to Alzheimer's disease is just huge. I don't know if any of you have taken care of an Alzheimer's patient, but it's not for the faint of heart. And let's see, I believe. All right. Okay, Madison, do the slides move if I do that?
No.
No? Okay,
No.
let's try this. Try this again. The one thing about Zoom that I just find remarkably annoying. Okay, now do they move if I do this?
No.
No?
No.
Oh, bloody heck. All right, then we'll just do this. And now do they move?
Yeah, they do.
They do. Okay. Okay, we stopped. Yeah, we stopped right here, I think. Okay. All right. Guys, have a good break? Go anywhere interesting? Anybody? Florida? That's interesting. Ohio? I was in Ohio Saturday for a Bible quiz. I was in Holland, Ohio. Right on the edge of Toledo. Yeah. Sorry? Alaska. I think you probably win the interesting competition. Alaska. Do you have family in Alaska? Okay. Where in Alaska? Never heard of it. Okay. Construction stuff, running a camp, something like that, or all the above. Okay. Hard work? Yeah. First graders, they'll do it. Okay, so let's consider this particular test case. So, woman, 35 years old, so she's an older mother. That means her risk for certain types of genetic conditions are going to increase. As the woman gets older, her chances of non-disjunction go up. 35, that's pretty much the red zone where things really start to rise in terms of chances of non-disjunction. So her result is positive for Down syndrome. That indicates she has a 1 in 200 risk for a baby having Down syndrome. So 1 in 100 risk would be 1%. 1 in 200 risk is 0.5%. So this is a screening test. Screening tests ascertain risk. Do they tell you a yes or no question? Give you a yes or no answer? No. First thing you have to acknowledge, limitation of screenings. So it doesn't mean your baby has Down syndrome. So you have a 1 in 200 chance, that's a 0.5% that your baby has Down syndrome. That means you've got a 99.5% likelihood that the baby does not have Down syndrome. And so if you could use any sort of example you want. 200 women your exact age, one of them is going to have a baby with Down syndrome, the remaining 199 would not. Or you have 200 marbles in a jar and one of them has a black dot on it. And you pull a marble out of the jar at random, 1 in 200 chance you'll get that marble with a black dot. Or you take 200 3x5 cards and you put an X on one of them and you throw them up, wee, like that, and then you shut your eyes and you feel for a card. You have a 1 in 200 chance of picking up the one card that has the X on it. You know, whatever example you want to use, try to give people an example, a feel for what these chances are. Try to use some sort of very mundane example, everyday example that tries to, 200 women, one of them, whatever. Because numbers sometimes to people, they have a rough time putting them into context. Now, as we said before with the Human Genome Project, you can detect for sequences of genes. Sometimes that sequence information can be very useful. Other times that sequence information just tells you that it's different from the reference sequence. The reference sequence, however, may be completely normal and your sequence might be normal too. The other thing this doesn't tell you, it doesn't tell you the expression pattern of the genome. Now, there isn't a way to do that. There's a technique today called messenger RNA sequencing. Messenger RNA sequencing is now usually designated as that. This will tell you exactly what genes are expressed in that cell. It'll even tell you how much of each transcript you have in each cell. Very powerful technique, really expensive. As this technology starts to become more and more miniaturized, and as next generation sequencing gets cheaper and cheaper and cheaper, this is going to start to be covered by insurance policies. You're going to start to use clinicians using this more. This is extremely powerful technology. It's not yet available in the clinic, I have to say, just because of the cost. The cost is coming down, quickly. These types of sequencers used to take up a space in the lab like this. Now they take up a space in the lab like this. Pretty soon they're going to take up a space in the lab like this. The miniaturization revolution is continuing. All right, so whole genome sequencing now is available, and it's cheaper than it's ever been. Again, whole genome sequencing can tell you something. Microarray analysis is being replaced by messenger RNA sequencing, because in some cases you can have extra copies of genes. Those are called copy number variants. Are those copy number variants expressed? You don't know, because you have the sequence. You can start to see now why this type of analysis is actually more informative than just getting the sequence. All right, so whole genome sequencing is available, and it's even going to be ordered in some cases. Some insurance policies nowadays will cover it. You can get an entire human genome sequence in an afternoon now. Use a technique called 4-5-4 sequencing. 4-5-4 sequencing. It's a form of next generation sequencing. Next generation sequencing. It's usually abbreviated like that. All right, so the challenge comes when you get these things called variants of significance, or you get sometimes variants of uncertain significance. One of the best examples is with this breast cancer 1 and breast cancer 2 gene. That's the gene probably that's got the most data for it available. Let me give you an example of risk factors for people who carry certain alleles of BCRA1 or 2. So people with mutations in breast cancer 1, BCRA1, they have a 74% increased chance for breast cancer. They have a 44% increased risk for ovarian cancer. Breast cancer 2, BCRA2 carriers, those who carry mutations in BCRA2, have a 69% increased risk of breast cancer. They have a 17% increased risk of ovarian cancer. These particular alleles also tend to be found more prevalently in people of certain ethnic backgrounds. So for example, Ashkenazi Jews, Jewish populations, there's two groups of them, one from Eastern Europe, that's the Ashkenazi population. The other are the ones who came up through Spain. They're the Sephardic Jews. Sephard in Hebrew means Spain. That's Sephardic Jews. The Ashkenazi population carries BCRA1 and 2 at a frequency of about 1 in 400. Sorry, 1 in 40. In the United States, it's 1 in 400. In the Ashkenazi population, it's 1 in 40. If people are of Icelandic descent, 8% of female breast cancer cases in Iceland are due to these mutations in BCRA1 or 2. 40% of male breast cancer cases, yes, males get breast cancer, and 6% of ovarian cases are due to BCRA2 mutations. French Canadian. French Canadian. Anywhere from 5 to 6% of the French Canadian population carries mutations in BCRA1 or 2. Latin American, particularly Latin American populations in the United States. Have 5 times increased risk of carrying these mutations than non-Latin American populations. And then finally, African American populations. African American populations, 12% of people carry either BCRA1 or 2. So depending upon their ethnic background, they might carry mutant alleles of one of these 2 genes. That increases their risk of ovarian and breast cancer substantially. What if you have these genes sequenced and you get an allele, but you have no idea if that allele is associated with cancer because there's just not enough data? Then you have what's called a variant of uncertain significance. A variant of uncertain significance. Might you get more information later that will tell you this is now an allele that's linked to cancer or not linked to cancer? Sure. But when you have the data in hand, you just have to tell them, we don't know. It's a variant of uncertain significance. Frustrating, but that's the information you have and that's the information you pass on. So variants can be either benign. There's very strong evidence that the variant is not associated with any particular disease. Likely benign, you have evidence, but it's not strong evidence. You wish it was strong, but it's likely benign. Uncertain significance, that's the one we just talked about. You either really don't have any data or you have data that's contradictory. Likely pathogenic, there's evidence, it's not strong evidence though. It's associated with some sort of disease, cancer, malformation, whatever. And then pathogenic, yep, strong evidence, there aren't a lot of doubts in that case. Those are the classifications of the various variants. So it's very clear that genetics plays a role in lots of different human diseases. And so you're starting to see nurses who are now being trained in genetic counseling. And in some clinics, you'll have nurses essentially who actually do some of the genetic counseling. I think the big part, the big temptation in genetic counseling is to overstate what the evidence says. You just remember, you are interpreting data and you're telling the patient, you're telling the parents, you're telling the whatever. You let them make the decision. That's the hard part about this field. So as you look at these, you can see that there are certain genetic conditions that increase the risk of certain types of cardiac issues, so-called channelopathies. We have mutations in proteins that code for ion channels or things like that. Almost half of seizure disorders are associated with some sort of genetic variant. Neurology, this one's a little slipperier because the inheritance might increase your risk for certain types of genetic diseases, but the penetrance is poor in many cases. Ophthalmology, now you're on much firmer ground. There are mutations that are associated with certain retinal problems. These might arise early in life more likely they're prone to apply to arise later in life, and they can be heavily influenced by things like diet and healthcare practices and things like that. Smoking, for example, is just bad for your retina. Certain types of macular degeneration, for example, smoking will increase the tendency for that to appear later in life much faster. Same thing with exposure to the sun. People who live at lower latitudes where the sun is much more intense are much more likely to experience these types of retinal dystrophies and things like that earlier in life and what have you. And then, of course, oncology. Family history is really important in oncology. And then ear, nose, throat, that's mainly having to do with hearing loss. Some people tend to be more prone to sinus infections, the shape and the structure of the sinuses. Some shaped sinuses may drain more easily than others, and that's something that tends to be inherited as well. You'll kind of see that, I think, in your practice. You'll see sinus infections will kind of run in, the tendency for them will run in families and what have you. Yeah, you are very much the go-between between the lab and the provider who's ordering the test. Access to genetic counselors is not great in rural areas, but with telemedicine, that's going to change that. I was chatting with a gentleman who's a clinical psychologist. He says virtually all psychology now, at least half of their sessions now are on Zoom. So I have Aaron Becker shaking her head saying, I hate Zoom. I hate doing medicine by Zoom. It's the future. It's the future of medicine. And some of you, when you're on call, your job may be to man the telemedicine line. You'll have to get online with mothers going crazy because their kid won't stop crying, or some old chap whose rheumatism is acting up again, or maybe a young chap whose rheumatism is acting up again, or something like that, or somebody with a sinus infection. Oh goodness, can I please have some antibiotics? Well, how long have you had this? Oh, 5 days. No, I don't think so, because most sinus infections are viral, so on and so forth. When should a person go seek a genetic counselor? You guys can read. You don't really need me to read these to you, but if you have essentially birth defects that run in the family, and if there's a history of unexplained death, that's really the big one there. The rest of these kind of really stem from those. Hearing, of course, hearing or vision loss as well, what have you. I'll let you read that. Here's some resources if you want to learn more about genetic counseling. If it's something that tripped your trigger, you might want to get into this. Go to some of these websites here, just take a look at them, and you might find them interesting. Or if you end up doing family medicine or pediatrics and something comes up and you want more information, these are all really good sites to take a look at. So the big, I think, take home of this chapter is watch this space, because as we learn more about the human genome and as more genetic mutations become identified in genes, we're going to have more ability to identify certain pathogenic variants. Because of that, there'll be less uncertainty when you get sequencing sequence data. And also it will, I just think this is going to start to come now to the small clinic and what have you, because the tests will become cheaper and the ability to identify things will become more straightforward. Blah, 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 and the punchline is. All right, are there any questions about that chapter before we move on to neurodegenerative conditions? All right, I'm not Okay, so our goal is to take a look at Alzheimer's disease, Huntington's disease, take a look at the genetics of these two conditions and what have you, and try to make some sense of them. Just tell me what you think, what you've heard about Alzheimer's disease. What do you know about Alzheimer's disease? Just give me some shout outs here. Okay, they have plaques, these amyloid plaques. Do those cause Alzheimer's? They might. Do you know definitively if they cause Alzheimer's? No. No. There's good correlation with Alzheimer's, but do they cause it? We have to honestly say we don't know. So you get neurofibrillary tangles, amyloid plaques. What happens to the gray matter in general of the brain? Shrinks, atrophies, good. Behaviorally, what do you see in Alzheimer's patients? Sorry? Memory loss, especially short-term. Long-term tends to be preserved for a longer period of time. That goes too eventually. They can become agitated, particularly in males. Yeah, agitation. They get confused. When they get confused, they can get belligerent. They don't know where they are. Who are you? What am I doing here? Where's my, whatever, so on and so forth, blah, 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 something like that. They can become agitated and very difficult and sometimes dangerous to care for. And let me just say this, especially since everyone in this class is female, your safety needs to come first when you're on the job. You should not be by yourself with someone you know is going to get agitated and, oh, they're older. It doesn't matter. When they get agitated, they can get that adrenaline hit and they can hurt you. So make sure you have help, essentially, if that sort of thing does arise. Right, so you can become agitated. What else? Struggles to talk, struggles to put sentences together. Sundowning. What's sundowning, Rebecca? They get worse. The sorts of things that are manifesting, the symptoms, the signs, they get worse as the sun goes down. Sundowning is very common. Very common. And sleep. What does Alzheimer's do to sleep? They sleep better or they sleep worse? Sleep worse, a lot worse. So your reaction might be, well, they're sundowning, just put them to bed. What happens if you put them to bed? They're going to get up and wander around because Alzheimer's disrupts sleep. It disrupts REM sleep, especially. It disrupts deep sleep, and that can cause their behavior to get even worse. Disrupts sleep, affects memory. They can get agitated. What else? Sorry? Eating. What about eating? They have trouble with swallowing. They can have trouble taking in nutrition. Some of them, really late stages, they'll just get in a fetal position, essentially. And they'll be fed usually through a G-tube, usually under those types of conditions. Usually not a central line, usually just a G-tube. Usually will be fine under those types of things. So it's, acalculia. Acalculia just means they can't calculate things or inability to, I have 3 apples and 2 in this bag. If I put 3 apples in this bag, I'll have 5 apples. They just can't do that. My mother was one of the first female brokers for E.F. Hutton. She worked in the LA stock market. In those days, you didn't have electronic readouts. You'd have people who'd go up and would hang the numbers and change the numbers. The little gaucho skirts that used to be popular back in the early 2000s that none of you wear anymore. Those were actually designed for the gals who were up changing the numbers. That was so to prevent you from flashing the world essentially while you're up on this rail. She was actually one of the gals who changed the numbers. She had to keep numbers in her head. Mom could do 3-unit multiplication and division in her head. I sat in the doctor's office and the doctor said, can you subtract 7 from 93? My mom couldn't do it. She couldn't do it. I left the room and I wept. I just wept. The lack of visual spatial orientation, confusion, and risk factors, of course, increased age, that's the biggie. Female gender, unfortunately. Low education level and positive family history, it does tend to run in families. There are things that slow Alzheimer's, it seems. Certain types of NSAIDs, in particular aspirin, low-dose aspirin. Statins seem to decrease the onset of Alzheimer's. Moderate ethanol intake, whether it's the ethanol or it's something in red wines or something like that, hard to say, but it's clear that people who drink low to moderate amounts of certain types of at least colored alcoholic beverages. I'm not sure it's been studied with colored grape juice or anything like that, but. And then social support network. How many of you are familiar with the NUN study, N-U-N? So the NUN study, there was a group of Carmelite nuns who lived together in a convent. These were ladies essentially who took their vows when they were your age, essentially, and they spent their days serving the community, gardening, praying, reading scripture, meditating on it, sleeping, getting up, doing all the above, so on and so forth. And they had this tremendous community, this constant exercising of the mind with spiritual disciplines and social support. And if one of them got sick, the others would help care for them, and so on and so forth. These nuns agreed to have their brains examined after death. Psychoneurologists wanted to know essentially what this kind of tight support network did for the brain. When they examined the brains of most of these nuns, they discovered many of them had neurofibrillary tangles and plaques in their brains, yet none of them showed the signs of Alzheimer's. It seemed as though the brain, with its constant working, was somehow able to grow and repair itself and work around these plaques. It's a remarkable study. It was in National Geographic 2006, I think, or something like that. Don't quote me on that, but there's 2000 something, the NUN study. Big study, very interesting. Social support networks are huge, especially for people with Alzheimer's. It's very clear that the degree to which someone declines, the speed at which they decline, is delayed by active social support network. That's why putting retired people in homes and then ignoring them is one of the worst things you can do for them sometimes. So early symptoms, you have short-term memory loss. So aphasia, oh goodness, what's that word? Agnosia, the inability to smell, difficulties with executive functions and so on and so forth. Things they could do easily, now they can't do. Later then you start to see personality and behavioral changes. Hallucinations too, pretty common. The hallucinations may be benign. Sydney, that cake you brought me yesterday was delicious. And Sydney goes, what cake? Furthermore, I don't bake. No, you probably bake. You bake? Sometimes, okay. That cake you baked for me was terrific. You're just, cake? Bake? I'm at nursing school. I don't have time to bake. So it gets benign. Oh, Madison, you visited me yesterday. We had such a good talk, didn't we? You're sitting there going, sure. I was in Florida yesterday. So they can be benign at first, and how you deal with a patient who's going through that. My grandmother, for example, with a name like Baratovich, my heritage is Croatian. My grandmother, now her English was pushed out of her head. She spoke nothing but Croat. And my Croat is not very good, but I understood when she said, I killed a chicken for dinner and I don't know where I put it. I looked at my dad and I said, Dad, did I get that right? Chicken? Kill the chicken for dinner? And I lost it? Dad said, yeah. I said, Dad, what do I tell? Dad then tells her, oh, it flew away. And my grandma goes, oh, yeah, okay. And Dad said, she's on the farm. We had chickens. She used to go out back and then pluck the chicken and cook it for dinner. So she's now become displaced in time and space. Mutism. What's mutism? Not talking. And then later, as Bree said, difficulty eating, swallowing. How do you do a swallow test? Just without calling a speech therapist to do a swallow test. How do you do a swallow test? No. How do you do a swallow test on someone? You go ahead, put your hands just right here and swallow. Did you feel how it went like that kind of? Tense, relax, tense, relax, tense, relax, tense, relax. That. Go ahead, do it again. Feel it? That wave that went down. You just essentially put your hands right here. You don't need to choke them. Just lay your hands there and say, give me a swallow. Give me a swallow. And they swallow. If you feel the wave, essentially, it's normal. It's normal. Now, stuff may get stuck. You might need to mess with their food, need to mix it. And a speech therapist usually, nurses used to do swallow stuff. Now speech therapists do that. You call a speech therapist. They do the swallow test and they will essentially determine what food works. And then if they get to that point where they just can't take in nutrition, then it's time to do a G-tube. All right, there's two main types of Alzheimer's: early onset, late onset. The early onset is inherited, strongly inherited. Early onset is also not nearly as common as the late onset. So the early onset counts for less than 5% of the cases, but it tends to occur before the age of 65. It can even occur as early as 30, age 30. So there's three main genes. They're on chromosomes 1, 14, and 21. They are inherited as autosomal dominance. Autosomal dominance. All right, now I need to explain this to you, I need to go into a little bit of cell biology. So you're going to need to just stay with me here. This is complicated, but it'll, I think, help you perhaps understand. Now, on the surface of neurons, you have this protein called the amyloid precursor protein. We're going to abbreviate that APP. APP. Amyloid precursor protein. What does it do? Not terribly clear. Most of the work done on it suggests that it's involved with helping neurons early in their development find where they need to go. So it's involved with neuronal honing. As you can see, amyloid precursor protein is inserted in the membrane and it's processed. It can be processed in one of three ways. This way, and this way. Now, this alpha cleavage site, it's cleaved by this group of proteases that are in the ATOM group. That stands for a disintegrin and metalloprotease. It's a large group of cellular proteases. This one just clips it, boom, right in the middle there. That peptide doesn't give you any trouble. There's a second way to process it, and this is called the beta secretase. Beta secretase, boom, clips it right up there. That little peptide there doesn't give you any trouble. Third way you process it is after the beta secretase processes it, you get another clip right here, and you make this little piece here that's called A beta 42. A beta 42, it's 42 amino acids long. A beta 42 forms the plaques. You can artificially express that in mice, and their brains will fill up with plaques, and the mice now will not remember how to go through mazes. And if you do the water test where you put them on a, you take a mouse and you put them in a tank of water, there's a platform, and then you just kind of, you brush them off the platform, then you pick them up and you put them on the platform. You do this in the dark. You brush them off it, and then you put them back on the platform a couple of times. That way they learn. You always brush them off the same way. That way they can swim and find it. If you do that to a normal mouse 3 times, every time they'll know how to get back to the platform. You do that to a mouse, or if you express that, the mouse will help, 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 glug, 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 and they won't be able to find their way back to the platform. So learning and memory severely inhibited in those mice. This particular protease is called the gamma secretase. The gamma secretase is a complex. It's a complex of several different proteins. Now, I threw this in here just to show you where essentially in the protein you're doing these cuts. So can you see this all right here? All right, so right here, this is the beta site here, that's the alpha site here, and then this is the gamma site here. So the beta secretase will clip this protein here, and then the gamma clips that there. So if you clip here and here, this little piece here, see 1 to 42, that's the A beta 42 piece. That's the piece that causes amyloid plaques. If you clip it here or here, you don't get that. And if you do clip this here, that piece there is not going to give you the plaques. So you clip here and here, and you get this piece there. That's what's going to cause you to get the plaques. All right, you with me so far? So here are the components of the gamma secretase. There's several proteins there. So in the red, we have a protein called nicastrin. Nicastrin, as you can see, is the biggest protein in this group. This protein here in the yellowish orange, that is presenilin 1. Presenilin 1 is one of those proteins that if you have mutations in it, it increases your risk of familial early onset Alzheimer's. Presenilin 1. The other proteins in the blue over here is a protein called PEN2. And then the green over here, you have this protein called APH1. So all together, these 3 proteins form your gamma secretase. This was a crystal structure that came from this paper published in 2015. All right, so mutations in either presenilin 1 or presenilin 2 increase your risk of early onset Alzheimer's. Likewise, mutations in the nicastrin gene can also increase your risk, as can mutations in the amyloid precursor protein gene. All of those can increase your risk of Alzheimer's. The APP gene is found on chromosome 21. Presenilin 1 is found on chromosome 1 Presenilin 2 is found on chromosome 14. What about late occurring Alzheimer's? You tend to get these symptoms after age 65. There are alleles of ApoE, which is on chromosome 19, that can increase your risk. There are alleles of ApoE that are known to increase your risk. Again, notice the note here, this is important. 1 or 2 copies increases your risk, but it doesn't guarantee it. Now, this is largely work that comes from animals, but preliminary work in humans suggests that this is the case. Amyloid plaques, when they first start to form, they can be removed. Just guess what helps remove them. No, actually. Sleep. Sleep. When you look at the brains of animals that are sleeping, the blood vessels dilate and the exchange of the cerebrospinal fluid increases. That's the time when your brain is cleaning itself. That's why virtually every early onset neurodegenerative disease is linked to a sleep disorder. And there are certain things that are known to protect the brain, and there are certain genes that are listed here that are known essentially to help protect the brain against the ravages essentially of amyloid plaques. Some people think everybody gets amyloid plaques. Some people just are better at getting rid of them. It's when they accumulate to a certain level, and then they'll accumulate to a certain level where they even disturb sleep. So your ability essentially to get rid of them now goes away as well. And the amyloid plaques then lead to these neurofibrillary tangles. And those neurofibrillary tangles are very rich in a protein called tau. Tau is a protein that organizes microtubules. They're very important for supporting the axons of neurons. And this essentially is what you get with a amyloid plaque. The material in that amyloid plaque, if you give it to cultured neurons, the cultured neurons die. So A beta 42 is very toxic to neurons. Some of the death is probably due to something called excitotoxicity. Dying neurons sometimes release all these stimulatory neurotransmitters. They overstimulate certain neurons, and essentially the neuron dies because it gets overstimulated. Here is a neurofibrillary tangle. Here it's stained for the tau protein. And essentially these axons now, the tau just gets all tangled up. Tau in these tangles is also phosphorylated. That seems to play a role in its accumulation in the tangles. And then, of course, brain atrophy. You compare an Alzheimer's brain with a healthy brain. I think that picture really says it all, doesn't it? When is diagnostic testing warranted? Not for late Alzheimer's. The association between ApoE alleles and late Alzheimer's is just not tight enough to warrant it. All you're really going to do is cause someone to worry. You want your patient to worry? No, you want them to live life to the fullest. So ApoE testing is not warranted. Early Alzheimer's is. Early Alzheimer's is. Now, remember, you can't really tell if someone has Alzheimer's while they're alive. It's only with a post-mortem autopsy that you can really tell if someone's got plaques and tangles and things like that. We don't have good stains and imaging that can see the plaques essentially on brains. Most of the protocol nowadays comes from symptoms and family history and neuroimaging. Do you see atrophy of certain parts of the brain and so on and so forth? Because learning and memory is dependent upon cholinergic neurons, using medications that increase the concentration of acetylcholine in the brain can delay, but not arrest, the decline of early Alzheimer's. So there are these drugs that are acetylcholinesterase inhibitors. Acetylcholinesterase is the enzyme that degrades acetylcholine by increasing local concentrations of acetylcholine by blocking the degradation of acetylcholine. You can increase learning and memory. These drugs will usually work for a while and then they will stop working. Also, these drugs are cholinergics. You're going to have side effects with these drugs. What kind of side effects are you going to get with cholinergics? Acetylcholine, think where it's used in the body, what it's used for. What group of neurons peripherally use acetylcholine? Think autonomic nervous system. Do sympathetic nerves use acetylcholine? Do parasympathetic nerves use acetylcholine? Okay, starting to see some nods. Postganglionic parasympathetic nerves use acetylcholine. Now, what sort of tissues are innervated by parasympathetic nerves? Sorry? Yes. So with more acetylcholine, you're going to get more or less motility in the gut. More diarrhea. Diarrhea is going to be a genuine side effect of these medications. Saliva. Does acetylcholine help with saliva production? So you can get drooling. Drooling as well. How about the heart? What's going to happen to the heartbeat? It's going to go down. Bradycardia. That'll be another side effect. So these are all side effects you're going to have to manage. Donepezil, the trade name, I believe, is Aricept. Rivastigmine, Exelon is a trade name, I believe. Galantamine, I forgot what the trade name for that one is. Now, there's another drug that's used. It is a NMDA receptor antagonist. Some of the agitation seems to, and some of the nerve degeneration, this excitotoxicity, seems to be glutamate linked. Blocking these receptors with a drug like memantine seems to help. And memantine plays well with these drugs here. Namenda is the trade name of that drug. I can't emphasize this enough. While these drugs can help, a positive social structure actually helps just as much. And if you put that on top of these, you can greatly increase the quality of life for an Alzheimer's patient. Any questions about Alzheimer's, the genetics of it, before we move on to Huntington's. Great, yeah, Macy. It can, yeah. Yeah, incomplete penetrance. Alzheimer's influenced by lots of other things. Now you're talking about late or early onset. Late onset can skip generations. It can skip multiple generations, in fact, sometimes. If it's in their family, if other people in their family have suffered from it, then that's a sign essentially that the risk is increased. But there are lots of other lifestyle choices that seem to influence the onset of Alzheimer's as well. Uncontrolled hypertension, uncontrolled high cholesterol are two big factors that contribute to Alzheimer's. What sort of mental life does someone have? It seems as though someone who's someone who reads a lot and who uses their mind a lot, that they seem to be at lower risk for Alzheimer's than those who, watch YouTube all day or something like that, or what have you. YouTube probably rots your brain from the inside out in the first place. Anyway, so yeah, it can, because it's influenced by so many other things. And that's why the ApoE allele testing is just not recommended. Huntington's chorea is an autosomal dominant. This one will not skip generations. This disease is relentless, it's progressive, and it's irreversible. You start out with so-called chorea, uncontrolled movements. You get sometimes a writhing. If you ask someone to hold, they might writhe. That's called atheliosis, the writhing sort of movements. You might see other types of tics, things like that. There are amyloid-related deposits in the basal ganglia. The big issue here is the death of GABAergic neurons. And the patient usually will progressively decrease, and it'll take 15 years. It's not a very pleasant way to go. Again, more cell biology. So Huntington, the protein involved with Huntington's disease, what it does is not completely clear, but you get in this protein, you have essentially this codon, CAG. It's repeated several times, and this encodes for the amino acid glutamine. And the single letter code for glutamine is Q. So polymerases, when they get to these stretches of CAGs, they will undergo polymerase slip. And whereas you might have anywhere from 8 to 12 of this polyglutamine stretch, polymerase slip can now cause this to go up to 24 to 36 amino acids in that stretch, glutamines in that stretch. And then if they get even higher, 40 up to 60 or something like that, this protein now is going to clump with other types of proteins that are necessary for the cell to do its job. It's going to grab those proteins, pull them out of solution. You're going to get a structure that forms in the nucleus of the cell. It's an intranuclear inclusion, sometimes called a Lewy body. This causes the death of the neuron. You have lots of problems with misfolded proteins and things like that. This is an example of a so-called triplet expansion disease. Triplet expansion disease. Huntington's is the example that's the best characterized of these. There are a few others. Okay, we are out of time. We'll finish this, and then on Wednesday, we'll go to the next chapter. All right? And I'll see, well, a lot of you tonight. You can turn it in electronically, or you can turn it in just to me, hot little hand. Either way. Thank you. Oh, look at that. Different colors. Oh. You can turn it in tonight. Yeah, that's fine. That's fine. Yeah, sure.
Mine's all stapled together. My back.
Perfect. Okay, lovely.